Go to cloud. Sorry. All right. So in every lecture, there's going to be, I've penned out some learning objectives. Please look through these while you're studying for either the quiz next week or the final exam in 2 weeks, I think it is. This is, if you master these, you will probably do very well on the exam assessments, because this is where the questions are going to come from. All right. All right. So first of all, I'm going to try to talk a little bit about why are we studying endocrine physiology? Why is it necessary? All right. So for maybe you don't realize, but endocrine diseases are very common in the general population. They can sometimes affect up to 10 to 15%. And they're always going to be, you're going to repeatedly see these in the clinical situations. Right? So the best example is diabetes mellitus. So the inability to respond to insulin correctly. So this affects about 1 in 10 people in the United States, and that patient population is growing due to the epidemic of obesity. So as obesity prevalence increases, the rates of type 2 diabetes is also going to increase. Next week, we're going to talk about insulin signaling. We're going to tell you about how that's going to be in the endocrine pancreas section. There's also osteopenia, which is a low bone density. Now, one of the treatments for this is vitamin D. And next week, when we cover the parathyroid gland, we're going to discuss how vitamin D and parathyroid hormone regulate both calcium and phosphate metabolism and bone mineralization. And finally, there's inflammation of the thyroid, thyroiditis. This is caused by autoimmune disease. We'll discuss that in a little more detail when we go through the endocrine gland in lecture 3. So endocrine, there's also some very common diseases which also have an endocrine component to it. For example, arteriosclerosis, which is the buildup of fat and cholesterol in your arteries. All right, so next week we're going to talk about cortisol, what cortisol is regulated. So it regulates both glucose and lipid metabolism. And so you have dysregulation of cortisol, you can get high levels of cortisol in your blood, impaired glucose tolerance, which can feed back to help in the development of arteriosclerosis. There are also many psychiatric disorders which have an underlying endocrine component. For example, again, elevated cortisol. People with Cushing's syndrome often have panic attacks and depression. There are also people with hypothyroidism, so Graves' disease, we're going to talk about a little bit later. These people have, most of these patients have anxiety disorders and mania. And finally, there's hypothyroidism, which has an opposite effect. So you have attention deficits and depression. So what is endocrine physiology? So endocrine physiology, the basic function of endocrine physiology is to control whole body homeostasis. And it does this by coordinating the activity hormone signaling pathways that regulate different tissues throughout the body to maintain homeostasis. We have 3 components of the endocrine system. We have the endocrine glands. The main morphological feature of these glands is they're ductless. They secrete their hormone secretions directly into interstitial fluid or in the bloodstream. Then we have the hormones. These are organic molecules which are synthesized and secreted by the endocrine gland. And you can see they circulate in the bloodstream at very low levels, 10 to the minus 9th to 10 to the minus 12th. Those are nanomolar and picomolar levels. And when they reach their target cell, they're going to bind to a receptor. That receptor is specific for that hormone, and it's going to elicit some kind of a characteristic biological response. Again, this is high affinity. Even at 10 to the minus 9th nanomolar, that means you have 1 hormone molecule for every 50 billion water molecules. So this is a high affinity system. Right. The other thing about the endocrine signaling is that it's a broadcast system. So it has the same hormone that regulates multiple tissues throughout the body. So it's pleiotropic responses. And this is best characterized by these adrenergic receptors I'm showing you here. In this particular cartoon, you see that each one of these different receptors can bind to the hormone, but they're coupled to different intracellular signaling pathways. So we have the beta 1 and beta 2 receptors. These couple to the G alpha S or stimulatory subunits. They're going to activate adenylate kinase, make more cyclic AMP, and cyclic AMP is going to activate protein kinase A, and that's going to elucidate a biological response. Here we see that alpha-2 activates the G-alpha-I receptor. This is an inhibitory receptor. It's going to inhibit adenylate cyclase and decrease cyclic AMP production. And over here we have the alpha-1 receptors. These receptors couple to a completely different system. They couple to phospholipase C, which is abbreviated PLC. And when they activate this pathway, you're going to have an increase in calcium. So the bottom line is that the specific response for the hormone is going to be dictated by what receptor the different target tissues are expressing. And that's best going to be illustrated by this flight or fight response to the epinephrine surge. So if you see almost in a car accident, or you see a bear in the woods, you have this big surge of or adrenaline or epinephrine, and it's going to elicit a body response. So here we see in both the liver cell, which are called hepatocytes, and the blood vessel skeletal muscle, they both express a beta-2 receptor. But in the hepatocytes, this is coupled, the increase in cyclic AMP in this cell type is coupled to activation of phosphorylase. It's going to increase glycogen breakdown, which is going to increase hepatic glucose output. And now I have the sugar or the energy and metabolic fuel to run my skeletal muscles. In the blood vessels of the skeletal muscle, you see that that same receptor is coupled to a completely different biological response. Here, activation of adenylate cyclase is going to cause vasodilation. Now I'm going to shift more, I can shift more blood to my skeletal muscle, and I can run or fight or whatever you need to do to get rid of that, get away from that bear. You see that in the visceral organs, like the intestine, express a different type of receptor. It's the alpha-1. Epinephrine binding to the alpha-1 receptor is going to cause an increase in calcium. That's going to cause vasodilation. So now I'm going to shift blood from my visceral organs, which I don't need to digest any food at the moment. I need to run or hide. It's going to shift it over to the skeletal muscle. And finally, in the heart, we have both beta-1 and beta-2 receptors. So epinephrine binding to those receptors is going to increase the rate of they increase the heart rate, increase the force of contraction. That's also going to cause vasodilation. So you see that this same hormone release acts on many different tissues, and their biological response is dictated by what receptor those tissues are expressing. All right, so I have some chats already. All right. So and I apologize, I don't watch the chats as well as I should. So if anybody online has a question, please feel free to stop me at any time. Anybody in the audience right here, if you have a question, it's very informal. If you don't understand anything, please stop and ask me at any time. All right. Now that disappeared. That's lovely. Oh, there it comes. Okay. So in this cartoon, I'm showing you the different endocrine glands and the hormones that they're secreted. I don't expect you to memorize all of these. We're going to go first. The ones I'm going to show you are important. First, we're going to go through the classic endocrine glands, right? Why is that? This thing works like a bug. All right. All right. First is the hypothalamus. Hypothalamus is part of the forebrain. It's going to release different release hormone releasing factors. And these hormones are going to regulate the secretion from the anterior pituitary. It also secretes different hormones directly into the posterior pituitary. This is called oxytocin, an antidiuretic hormone, but it's also called vasopressin. And these have direct effects on peripheral tissues. Then there's a pituitary gland. The pituitary gland receives input from the hypothalamus, and it's going to release a second hormone. And that second hormone is going to affect different peripheral tissues and also the different endocrine glands. And we'll go through that in a little bit in the second lecture. Here we have the thyroid gland. Thyroid gland sits along the trachea. It produces the thyroid hormones, thyroxine, T3, and also calcitonin. This is where we talk about this in the parathyroid hormone. This is involved in calcium phosphate metabolism. On the back side of the thyroid hormone, you have at the back side of the thyroid hormone, you have the parathyroid hormone. This is involved in making the parathyroid gland. It makes a parathyroid hormone. The parathyroid hormone is involved in serum and phosphorus metabolism. And we're going to talk about that next week when we cover the parathyroid gland. Here we have the adrenal cortex, the adrenal gland sits on top of the kidney. We're going to focus mostly on the adrenal cortex. We're not talking too much about the medulla. The adrenal cortex produces both the glucocorticoids and mineralocorticoids and the sex androgens. These are involved in regulating responses to stress, for instance, hypoglycemia, mineral balance, water balance, and regulation of metabolism. Then we have the pancreatic islets. These are the islets of Langerhans. They produce both insulin and glucagon, and these are involved, hormones are involved in the storage and utilization of metabolic fuels. And we'll cover that next week when we cover the endocrine pancreas. And finally, the class of plastic endocrine glands are the ovaries and testes. These control the sexual function, maturation, and development. So you guys should know where these different endocrine glands are, their basic functions for any quiz and exam we're going to have. There are also secondary endocrine glands. These are tissue organs that have their primary function is not to produce a hormone, but but they can produce hormones, right? So one is the heart, usually is used to pumping blood, but it can also produce a hormone called atrial natriuretic peptide. This is involved in vasodilation and in decreasing the cardiac output. We're going to discuss this as a counter-regulatory hormone to the renin, angiotensin II, and aldosterone pathway, and we'll cover that next week when we cover the adrenal gland. And there's a kidney. The kidney produces a protease called renin, and renin is involved in the activation of angiotensin. And this is important in the renin, angiotensin, aldosterone axis, and we'll cover that again when we cover the adrenal gland next week. Then there's the gastrointestinal tract, and the GI tract produces many, many hormones. Actually, most of the hormones you find first found in the gut were then identified in the brain. I'm not going to have you memorize all of them. The ones that are going to be important, we're going to cover in these particular lectures are glucagon-like peptide 1 and gastric inhibitory peptide. And those are going to be important when we cover the endocrine pancreas because they regulate insulin secretion. Finally, there's the liver. The liver, in response to growth hormone, is going to produce another hormone called insulin-like growth factor. And this hormone produces most of the somatic effects of growth hormone. So it's going to be the one that stimulates cell growth. Now, some hormones are not secreted as an active compound. They need to be processed by peripheral tissues into the active compound, and see some of the important ones. So we have the lung. This is important converting angiotensin 1 to angiotensin 2. We're going to cover that in more detail when we cover the adrenal, the hypothyroid gland. Also the kidney. The kidney is involved in the production of vitamin D. We'll cover that in more detail next week when we cover the hypothyroid gland. Finally, there are many tissues that are involved in the deiodination of thyroxine, the T3. This is the more active compound of the thyroid hormones, so it has to be converted to T3. They have the biggest effect, and many tissues are involved in that. And we'll cover that in lecture 3 when we cover the thyroid gland. So of all these hormones we're talking about, this particular table shows you the ones that are most important for maintaining whole body homeostasis on a daily basis. And these are the ones we're going to emphasize during these lectures. So we have the hormone, where it is, and its basic action. So we're going to cover the thyroid hormone, we're going to cover the glucocorticoids, the mineralocorticoids, vasopressin, parathyroid hormone, and insulin and glucagon. And these are going to be covered in different lectures, and you can look at this, you can read through and see that we're going to cover what actions are in more detail when we get to those lectures. All right. So how do hormones signal to each other? So classic hormone signaling suggests there's an endocrine cell produces a hormone. It's released into the circulatory system and it travels some distance to a target cell that expresses a receptor, it binds and elicits a biological effect. Examples of this are all the trophic hormones that are released by the anterior pituitary, such as thyroid stimulating hormone. Then we have paracrine signaling. This is next to Here we have the endocrine cell producing the hormone. It's not released in the blood system. It's into the interstitial space. It binds the receptor on the neighboring cell to elicit a biological response. So an example of this are somatostatin, which is produced by the delta cell in the endocrine pancreas. This inhibits the secretion of insulin from beta cells or glucagon from alpha cells, and we'll discuss that in more detail next week when we cover the endocrine pancreas. We also have autocrine signaling. This is cell signaling. Here we have the endocrine cell producing the hormone and also the receptor responds to that. So the hormone is secreted, binds the receptor, and elicits a biological response. Examples of that are like most cytokines and insulin-like growth factor. And finally, this is not very common. You have intricate signaling. Here, the hormone is not even secreted. It binds to an intracellular target to elicit a biological response. An example of that is angiotensin II. And finally, we have neuroendocrine signaling. In this particular mode of signaling, we have the neuron producing and secreting the hormone into the blood system, travels some distance where it meets the target cell, expresses a receptor. Notice that only the cells that have the receptor have a response. If it doesn't have a receptor, there's no response. Examples of that are the hormones vasopressin or oxytocin, which are released in the posterior pituitary gland. All right. So most hormones are classified by the chemical nature. And this is done because most hormones, if they come from their chemical nature dictates some of their properties, whether they're how they elicit a biological response, how they're transported, and how they're made. We have, in this particular example, we have the peptide hormones. Insulin, a very good example. We have the steroid hormones, who are all synthesized from cholesterol. These are the mineralocorticoids, the glucocorticoids, the androgen hormones, and vitamin D. You have the hormones derived from a single amino acid. We have tyrosine, which makes the catecholamines, epinephrine, dopamine, and norepinephrine. Tyrosine also the precursor that makes the thyroid hormones, thyroxine. So it's here, this is a single residue, tyrosine residue right here. And up here is the indole ring from a second tyrosine. We'll go through how that is manufactured when we look at the thyroid gland. You can also have hormones generated from tryptophan. This makes the neurotransmitter serotonin. And serotonin can also be converted into melatonin. Melatonin is secreted by the pineal gland to regulate circadian rhythm, but we're not going to go into much of that. We're not going to do all that much detail. Again, I don't want you to memorize the structures, just the basic categories. Right? In this chart, I'm showing you all the hormones that we're going to talk about in these lectures, and their different chemical natures. We have the steroid hormones. You see we have the sex hormones, we have the mineralocorticoids, the glucocorticoids, and vitamin D. In this particular graph, they're divided into small peptides and large proteins. These all have the same, very similar biological properties. They're all water soluble. They are transported in the blood system without any carrier proteins. They bind to extracellular receptors to elicit a biological response. The same thing for steroid proteins. So they're all made from cholesterol. They're all lipophilic. They can't be transferred in the blood very easily, so they're all bound to transport proteins. And then we have the tyrosine derivatives. We have the catecholamines, the epinephrine, norepinephrine, and dopamine. These again are all water soluble. They're transported in the blood without any carrier proteins. They bind to extracellular receptors to elicit a biological response. Now the thyroid hormones are quite a little bit different. They're not very soluble in water, and these are all transported by carrier proteins, which we'll talk in detail later on. All right. So the biosynthesis of hormones and their secretion. So like I said earlier, all the steroid hormones are synthesized from enzymatic modifications of cholesterol. They're very lipid soluble. They pass through membrane, therefore they're not stored because they can pass through a membrane, so they can't be stored in any secretory vesicles. Then we have the peptide hormones and proteins. These are all encoded by specific genes. They are transcribed and translated by the normal protein machinery. They can undergo extensive post-translational modification like proteolysis, glycosylation. They are targeted in the secretory pathway. They are going to be stored in secretory granules, and they're going to be released when you have a stimulus to a process called regulated excitosis. Then we have the catecholamines, norepinephrine, epinephrine, and glutamine. They're all synthesized from the amino acid tyrosine. They are also stored in granules, and they're going to be released when there is a stimulus on. And finally, we have the thyroid hormones. These are all synthesized from tyrosine residues. They're on this particular protein called thyroglobulin, which we're going to discuss in detail when we cover the thyroid hormone. And these hormones are released by the proteolysis of this thyroglobulin. So, I mentioned earlier, steroid hormones are not stored, but they're synthesized and secreted on demand. So since they can go through a membrane, you can't store them in a secretory vesicle. The way this happens is that you either have to increase the synthetic enzymes that produce that hormone, or the carrier protein involved in their transport through the body. In this cartoon, I'm showing you the various different hormones and cytokines and growth factors, which can activate either the increased expression of this protein called STAR, and STAR stands for steroidogenic acute regulatory protein. You either increase the expression, or you can phosphorate to increase its activity. This protein is involved in transporting cholesterol from the outer mitochondrial membrane to the inner mitochondrial membrane, and that is a rate-limiting step to producing all your steroid hormones, because it makes pregnenolone. So this is how most of the steroid. This is the key rate limiting step in the synthesis of all steroid hormone coming out of the adrenal gland. Now, go through a little bit about how peptide and peptide hormones are made. So, like I said earlier, they're all encoded by specific genes. That specific gene is transcribed into an mRNA, and mRNA is translated. There are signaling sequences that they target it to the rough endoplasmic reticulum. It starts to make a messenger RNA into the lumen of the ER. That signaling peptide that generates it is then cleaved off by a proteolysis, generating a pre-pro hormone. That prohormone also has signaling elements inside it, which target it to the Golgi apparatus, to the secretory pathway. You see it going down through here. In that secretory pathway, it can undergo other post-translational modifications, proteolysis, glycosylations. At the end of that, you see that we have the active hormones produced by this proteolytic cleavage. It may also be glycosylated. Maybe it forms disulfide bridges. But then a different way to do it. And when you have in the secretory vessel, you have the active hormone and the different peptide fragments, which are all going to be released at the same time. When you have a signal, either cyclic AMP increases cyclic AMP or increases in calcium, it's going to cause that vesicle to fuse with the plasma membrane. It's going to release that hormone into the extracellular fluid, ECF. Those hormones are going to then be taken up by the capillary endothelium, into the bloodstream, and then transported to any target, wherever they're going to find their target. Specific target tissues which have the expressed receptor for that hormone. Right. So here are some examples of the different ways proteins can be processed. This is a pre-pro hormone for a thyrotropin releasing hormone. As you can see, it's a very large hormone and has 6 repeats of the thyrotropin releasing hormone. You can see there's only 3 amino acids there. So all of this has been chopped up, and you get these are the hormones, and this is what releases. These peptides and the signal peptide fragments are also released when that secretory vessel is, binds to the plasma membrane and fuses and releases its content. You can also have glycosylation. So here are the different proteins, hormones, which are glycosylated, right? Thyroid stimulating hormone, follicle stimulating hormone, and luteinizing hormone. And this is just a schematic showing some kind of glycosylation, right? So glycosylations are important because you may have an alpha and beta subunit of the hormone, and the glycosylations are help it to bind together. Glycosylation also helps protect the hormone from proteolysis, right? And sometimes glycosylation is required for the hormone receptor to bind to that hormone. So it needs it for full activity. So I've made the hormone, I put it in the secretory vesicle, now how do I get it out? So most of these peptide hormones and catecholamines are released through a process called regulated excitosis to secretory vesicle. So in this particular process, you have a stimulus. It can either be a rise inside calcium, a multisensitive calcium channel, or release from internal stores, or an increase in cyclic AMP. That's going to be the signal for this secretory vessel to start transporting to the plasma membrane. And you can see it transports to the membrane on the cytoskeletal elements. These are either microtubular or the microfilament systems. Once it reaches the plasma membrane, that vesicle fuses. Now, there are going to be some other parts in there I'm not going to talk about, but the vesicle, this granule fuses to the plasma membrane. When it fuses, it's going to release that hormone content into the extracellular space. So now I have the hormone released. So how do I transport it? I got to get into that. So most of the peptide hormones and catecholamines are water soluble, and they circulate freely in the interstitial fluid in the blood. So you don't need any carrier proteins. The exceptions of these are going to be growth hormone and insulin-like growth factor, which are not very water soluble. And a lot of those hormones are about 50% of them are being transported around on carrier proteins. And we'll discuss that in a little bit later. However, both the thyroid hormones and the steroid hormones are very lipophilic. Right? They do not dissolve in water or blood plasma, and they have to be transported around the blood system on carrier proteins. In this graph, I'm showing you the specific transport proteins and the hormones they're carrying, right? So realize that most of these globulins, all these globulins are made by the liver, and they have a very high affinity for the individual hormone that they're going to bind. So the examples here, we have corticosteroid binding globulin. This is going to bind our glucocorticoids and mineralocorticoids. We have thyroxine binding globulin. They're going to bind the thyroid hormones. And we have the sex hormone binding globulin, which are going to bind testosterone and estrogen. Now we also have other proteins which are very abundant in the blood system. They have very low affinity. The most important of these is going to be serum albumin. And since it's such a high concentration of this protein, it can bind some of these lipophilic proteins at high levels. And so also be very important in carrying the hormones through the blood system. Now, why is this important? So first concept of this thing. So it's only the free hormone that's going to bind the receptor and elicit a biological response. If it's bound to carrier protein, it is not going to. So the pool of free hormone is dictated by the rate of hormone secretion from the endocrine cell, the rate of degradation, and how much is bound to the carrier protein. So the carrier protein is very important in dictating the free to bound concentration of the hormone. Since the carrier proteins also circumvent the water solubility problem with these steroid hormones. It binds to them, it makes it more soluble in the blood system. So they're able to carry them. So they don't have to worry about it. Otherwise, these lipophilic hormones will all end up in fat and do us no good. By being bound to that carrier protein, we're going to produce a large reservoir of hormone that's able to be transported throughout the body. It acts like a buffer. So when a hormone binds the receptor, this concentration may decrease slightly, but then there's going to be some hormone come off the carrier protein to replace that free hormone, maintaining the concentration of the free hormone. So it's going to minimize our minute-to-minute fluctuations in free hormone concentration. Right? Secondly, when the hormone's bound, when the hormone's bound to the carrier, it can't be metabolized. It can't be attacked by proteases, and it can't be filtered by the kidney. So it's going to extend the half-life of the hormone. Right. So I secreted the hormone, I got it into the blood system. Either it's being freely, if it's water soluble, it's freely transported in the blood. If it's hydrophobic, lipophilic, it's going to be transported by carrier proteins. And once now I have to carry it to its target tissues, and I need to elicit some kind of biological response. So hormones are going to elicit their biological response by binding to receptors. I have two types of receptors. I either have cell membrane receptors or nuclear receptors. The cell membrane receptors are going to the ones that bind all the water-soluble molecules, like the peptide hormones and the catecholamines. Binding to that receptor is going to elicit some kind of a change in the receptor, and it's going to generate some kind of a second messenger. And that second messenger is either going to change protein synthesis, phosphorylation, or gene regulation. Steroid hormones are freely soluble through the membrane. They're able to penetrate in it. They're going to bind to some kind of a receptor. That receptor is going to be able to transfer it into the nucleus and turn on gene transcription. So I always said that. So almost all the steroid hormones and thyroid hormones are going to exert their effect by binding to what's called nuclear receptors. So in this little cartoon, we're showing you the hormone, either the steroid hormone or the thyroid hormone. It's bound to its carrier in the bloodstream. There's going to be some of that hormone going to be in equilibrium with the free hormone. Once it dissociates from the carrier protein, it can cross the plasma membrane of both the capillary cell and the target cell. Inside the target cell, it can either bind to a receptor in the cytoplasm or a receptor in the nucleus. That binding of that hormone to the cytoplasmic receptor I forgot this. There are some, we're not going to worry about this. There are some evidence that some of these hormones can bind to cell surface receptors for rapid response, but I'm not going to cover that in these lectures too much. All right. So, oops. All right. So the binding of the hormone to the cytoplasm changes the conformation and allows it to be transported through the nuclear pores into the nucleus. Binding of the hormone to the nuclear receptor inside the nucleus changes the conformation and allows it to bind to these different steroid regulatory elements. These are elements within the gene that I could either activate transcription or decrease transcription of the gene. So if I activate transcription, I'm going to produce more messenger RNA. The messenger RNA is going to be transported out of the nucleus. I'm going to undergo translation. I'm going to make more protein, and I'm going to produce the biological response that steroid hormone is going to be doing. Now, for water-soluble proteins, hormones like peptide hormones and catecholamines, these are going to go through couples of different things called G protein coupled receptors or tyrosine kinase receptors. Again, the hormone binds to the ligand binding domain, which is in the extracellular portion of it. It's going to produce some kind of conformational shape in the protein. I'm either going to activate protein phosphorylation, or I'm going to change gene transcription and gene regulation. So all the short-term effects are going to be brought about by altering cell protein function. This is usually through phosphorylation, like I said, mostly. It's the most common post-translational modification. And the long-term effects of these hormones are going to be through changes in gene expression. All right. So here, I'm going to show you examples of all the hormones that use the cyclic AMP mechanism to control cell function. We have the stimulatory ones and inhibitory ones. The ones that activate adenylyte cyclase activate this protein called G alpha S. It's a stimulatory, like I said earlier. It's going to bind to the adenylyte cyclase. We're going to produce more cyclic AMP, and we're going to raise cyclic pKa activity, and that's going to be the protein that elicits the biological response. Alternatively, if the hormone binds receptor coupled to G alpha I, which is inhibitory, it's going to inhibit adenylyte cyclase. We're going to decrease cyclic AMP production, and I'm going to decrease PKC activity, and I'm going to inhibit that pathway. And here, I don't expect you to memorize all these. If I'm talking about a particular hormone during the lectures, you can refer back to this and see what signal transduction pathway it is using. All right. There are also G protein coupled receptors coupled to phospholipase C. In this particular schematic, showing the binding of a peptide hormone to its receptor, activates a G protein. This is called the G alpha Q, which is going to activate phospholipase C. Phospholipase C is an enzyme that cleaves a lipid molecule called PIP2, which stands for 4,5 phosphatidyl 4,5 bisphosphate. It's going to cleave that molecule in two. It's going to produce two second messengers. One is a lipid portion called diacylglycerol. The other is this thing called a sugar called inositol trisphosphate. IP3 is water soluble. It floats in the cytosol. It binds the receptor on the endoplasmic reticulum, and that receptor releases calcium from the ER to produce a calcium response, rising calcium in this and response. Diacylglycerol can bind to a different kinase called protein kinase C. It's going to activate that kinase. It's going to phosphorylate targets and produce a biological response. And here again are the different hormones which use this particular transduction system to regulate cell function. Again, I don't expect you guys to memorize this, but if I talk about one, you can refer back to this, see what kind of signal transduction pathway it's using. Then we have the receptor tyrosine kinases. We have the tyrosine kinase or receptor-linked tyrosine kinase. Here we in this particular example, these tyrosine kinases have an intrinsic tyrosine kinase domain. So when the hormone binds to the ligand binding domain up here, it's going to activate that tyrosine kinase domain. That tyrosine can autophosphorate the receptor itself. Autophosphorylation is going to produce these phosphates, which are like little signaling molecules. It's going to recruit docking proteins, are also called scaffolding proteins, and I'm going to build up a signaling complex and phosphate downstream targets. Examples of this are insulin and insulin-like growth factor receptors. We also have receptor-linked kinases. Here, the kinase is not part of the receptor, but is bound to it. These are tyrosine kinases. These are from the Janus family of proteins. So binding of the growth factor here sends a signal down to activate the receptor. These receptors usually phosphorylate a transcription factor called STAT. This stands for signal transducer and activator of transcription. And this is a protein that's going to go into the nucleus and activate change gene transcription. So examples of this are growth hormone and prolactin receptors. Right. So I've made my hormone. I've secreted it into the extracellular media, transferred it in the blood. Now I've bound to my receptor and I've generated a biological response. So once I've generated my biological, I need to get rid of that signal. I don't need it any longer. So these are usually done most importantly in the liver, either through phase 1 or phase 2 reactions. Recall that phase 1 reactions are through dehydrogenases, oxidases, and reductases. So I'm going to change the oxidation of double bonds in those molecules. Phase 2 reactions are the conjugation reactions. I'm going to conjugate more water-soluble molecules to the hormones to allow them to be excreted more easily. The most common one of these is sulfonates or glucuronic acid. So these things can also undergo productivity in the kidney. So those hormone metabolites are predominantly excreted in either the bile or the urine. Right? And because these are the two organs are the most important for hormone degradation, if you have liver disease or kidney disease, this can affect the degradation of these hormones in some cases. All right. In this chart, I basically summarized everything I just talked about. I have the peptide hormones, the steroid hormones, thyroid hormones, the catecholamines. I tell you what their mechanism of secretion is, if they're bound or not bound to carrier proteins, their lifetime in the blood. Notice that if I'm bound to a carrier protein, I last a lot longer. If I'm not bound to a carrier protein, I last very little. So the carrier protein prolongs life, the half-life of the hormone. If I have a long half-life, my mechanism of action is very long. If it's short, it's short. All right, so this is for you to study for any quiz. Here I tell you about the receptors, where they're located, whether they're plasma membrane or nuclear, and their mechanism of action. All right. So how do we regulate hormone secretion? So there's several different feedback mechanisms. In this particular example cartoon, I'm showing you the positive feedback mechanism. These are explosive and self-reinforcing. In this particular example, we have the endocrine cell. It produces a hormone A, which acts on the target endocrine cell to produce hormone B, Hormone B may have a biological effect, but it also feeds back on the endocrine cell to produce more A. And the best example of that is the feedback of estradiol back on the pituitary gland to increase luteinizing hormone and follicle-stimulating hormone secretion. And this is required for the rapid rise in these two hormones during the menstrual cycle. Now, these are not very common. There's not very many of these positive feedback loops. More common are the negative feedback loops. These are the most prominent mode of regulation. In this particular regulation, we have an endocrine gland, which produces a hormone, has some kind of effect on the target cell. Whatever the target cell produces a response, and that response is going to feed back and inhibit the endocrine gland. Right? This is preferred because it basically maintains stability in the system and maintains that whatever biological effect we're looking at in the normal range. So here are some examples of that. In this particular graph, you see that we're looking at glucagon secretion versus blood glucose concentration. You notice as blood glucose concentration decreases, as blood glucose concentration increases, glucagon secretion decreases. So in the alpha cells in the pancreas produce the hormone glucagon. Glucagon goes to the liver. It stimulates the release of glucose, increases hepatic glucose production. That rise in glucose is going to feed back and inhibit the alpha cell. Other examples of that are different levels of serum calcium regulating the secretion of parathyroid hormone. And we'll discuss that next week when we discuss the parathyroid gland. On this cartoon, we're showing the multi-level feedback mechanism between the hypothalamus, the anterior pituitary, and the target endocrine cell. And the hypothalamus is going to produce and secrete a releasing hormone. It's going to act on the anterior pituitary cells. And that hormone is going to stimulate the release of a tropic hormone. That tropic hormone is going to release into circulation and going to bind to a target cell, a target endocrine cell, and produce a biological response. Usually, it's going to either increase the proliferation of this particular endocrine cell, but also make it stimulate the production of an endocrine gland hormone. Now, there are many different feedback loops here. We have what's called a short feedback loop. This is when the trophic hormone feeds back and inhibits the release of the releasing hormone at that level of the hypothalamus. We also have these what they call long loop feedbacks. Here, the endocrine gland hormone is going to feed back, inhibit the release of the trophic hormone, and they're going to feed at the level of the anterior pituitary. It's also going to feed back at the level of the hypothalamus and decrease the secretion of the releasing hormone. And here are some examples of different hormones that use this feedback mechanism. We're going to talk about the thyroid hormone in a few minutes. Next week, we're going to talk about the cortisol with the adrenal gland, and also sex hormones use the same feedback mechanism. All right. This is a very busy slide. I'm not expecting you to memorize all the different patterns here. The take-home message is that hormone secretion is rhythmic. It can either take a pulsatile, for instance, luteinizing hormone. It goes up and down, right? It can follow some kind of daily pattern. For instance, testosterone shown here, it goes down during the day, decreases until your deep sleep, and then rises as morning arrives. Other examples of this that follow the circadian rhythm, cortisol and adrenal-corticotropin hormone, and some hormones are activated by sleep, for example, growth hormone. Other hormones are episodic, so there's some kind of episode. So in the example here, we have a infant suckling. When it suckles, it stimulates the release of oxytocin. Now, right? So this cyclic pattern of hormones is important for maintaining normal endocrine function. So if I was a doctor and I'm going to inject it, they say there's a hormone dysfunction, there's a less hormone. If you just inject a hormone, and get a bolus of hormone, that's not going to produce the same kind of biological response as these things where they're going up and down, right? So it's dynamic. So you can't just inject. You have to try to match what's going on naturally with the hormone. Second, if I'm an endocrinologist and I have a patient who has an endocrine dysfunction, if I just take a single blood draw, it's not going to be very informative because I have all these things going on. So what happens is that what you need to do is understand the dynamic regulation. A good example of this is growth hormone, right? So it goes up and down cyclic all the time. But if you know that there's different inputs can regulate that. So hyperglycemia, high blood glucose, suppresses growth hormone. Whereas certain amino acids like branched chain amino acids, arginine and lysine, stimulate cellular secretion. So if you know that kind of feedback regulation, you can challenge the patient with either, let's say, high glucose, a glucose pulse, and you can see how the growth hormone responds. So if it responds in the correct manner, you know that signaling pathway is intact. So I'm going to take a few minutes to talk about how we measure hormones. Right? In this particular example, I'm looking at a radioimmunoassay. This is a classic competitive binding assay. The key features of this is that we've generated some kind of antibody that specifically recognizes the hormone. We've also taken some of that hormone and we've radiolabeled it. So if you have a peptide hormone, it's very easy to add iodine, 125 iodine to the cell. If it's for instance, cortisol, you can actually radiolabel it with C14. So it's radiolabeled. If you just have those two components, that hormone is going to bind to the antibody, and you're going to have a very high level of radioactivity. But the more of the unlabeled hormone you add to it, it displaces that radioactive hormone from the antibody, and you're going to get decreasing amounts of radioactivity in your sample versus as you increase the unlabeled hormone. So it's very simple to say, okay, I've decreased the binding of the radioactivity by 60%. That means I have like 1 unit of hormone. So you generate a standard curve, and you're able to read off whatever you have in your sample. There's also another example of the enzyme-linked absorbent assay called ELISA. Here we have the peptide hormone, the antibody that recognizes the hormone. It's affixed to a plastic. It binds the hormone. Then we have a secondary hormone, which also secondary antibody that also recognizes the hormone. And it's coupled to something called horseradish peroxidase, HRP. This is a peroxidase, and it's going to use this substrate. It converts this substrate, which is colorless, to a blue molecule. You can see here, it's blue. And when I add acid to it, it's going to change it to a yellow color. And then by reading the absorbance, I can regenerate a standard curve. Here in this example, I have insulin. As I increase the concentration of insulin, I get an increase in the absorbance. And if I want to read off an unknown, all I got to do is measure the absorbance of the unknown, and I can read back and follow it back onto my standard curve. So determinants of hormonal activity. So these are kind of, I think it's fairly simple. So I've just penned down a few. So what determines the hormonal activity? So the concentration of the hormone at the cell surface. So that's going to be related to the rate of secretion, the rate of degradation, the rate of excretion, the rate of transport. We also have how much hormone is there, the duration of exposure. That relates to how long the secretion, how long the secretion from the hormone, the endocrine gland is going on, how much is secreted, the half-life in the blood, and how many pulses are going on. Like we saw with the luteinizing hormone, it goes up and down. What's the frequency of those pulses? We also have the competency of the target cells. How many functional target cells are there in the gland? How many receptors are on each cell? Is there been an upregulation or downregulation of the signaling molecules? There's also some effects of either antagonistic or synergistic hormones. For example, when we talk about the thyroid hormone, we know that thyroid hormone can feed back and inhibit its own secretion. It does this by downregulating the thyrotropin releasing hormone receptor in the thyrotropes. When we come to the thyroid gland, we also learn that the thyroid hormone has a major effect on the cardiovascular system. It does this by increasing the adrenergic receptors in the heart. So this is a standard, typical dose response curve. Notice the first thing is not an all or nothing response. Right? So it's graded. It's graded to proportion to the concentration of free hormone. And usually, if you plot the biological response versus a logarithmic concentration of the hormone, it's going to be a sigmoidal curve. And there are several key features on this graph that you think you should know. First of all, there is never a zero. There's always going to be some level of secretion, even if there's no hormone present. It's a basal level of hormone. Then we have the threshold. This is the level of hormone which I can actually measure a change from the basal level to an actual change in that biological response. Here's a maximal response. This is the largest possible effect that can occur with a saturating level of hormone. Now, this doesn't mean that all your receptors are bound. For instance, in some peptide hormone receptors, only 5 to 10% of those receptors are bound with hormone to get a maximum response. And we also have the sensitivity, so the IC50 or EC50, excuse me, which is also called ED50 or effective medium dose. This is a response, the dose of hormone that elicits 50% of the biological response of the maximum. Now, these hormone response curves can change in different ways. This particular one, I'm showing you changes that respond to the capacity targets to that. So this is done by either changing the number of receptors on the target tissue, how many cells are in the target tissue, or the rate limiting step of the intercellular pathway. And you see in this particular example, the half maximal effect is the same for each one of these dose response curves, but the maximal effect is different. And those differences are due to changes either in like I said, the number of target cells, the number of hormone receptors, or a change in the intercellular signaling pathways. For instance, you may have an upregulation of an enzyme that decreases cyclic AMP, so it degrades cyclic AMP. You can also have changes in the target sensitivity. So in this, let's say B is the normal. It's got an EC50 of 1 nanomolar. If I shift to the right, it's less sensitive. You notice that the peak is the same. I just need more hormone to elicit the same biological response. If I shift the dose response to the right, it's more sensitive. Now the EC50 is 0.1 nanomolar, right? So these things can be usually changed. The most common is a change in hormone receptor affinity. Or you can have a change in the rate of hormone degradation. So the hormone is being chewed up, so you need more hormone to elicit the same biological response. Right. So here are some of the mechanisms by which the number and affinity of hormone receptors can be modulated. So if you have a hormone receptor and it's bound to a molecule, if that binding is there for a long period of time, it can induce what's called internal receptor internalization. It can be internalized into endosomes, and when it's in there, it's no longer producing a biological effect. I can also have the receptor bound to the hormone for a long period of time, and that can trigger it to go into the lysosomes. And the lysosomes is the organelle which digests protein, so it digests that hormone receptor. I also, if I have the hormone bound to receptor for a long period of time, I can induce a phosphorylation receptor. And the phosphorylation receptor dissociates it from the downstream signaling pathway. A very good example is the BARC kinase. So if you have the adrenergic receptor bound to catecholamines for a long period of time, it's phosphorylated, and then you no longer produce any cyclic AMP production. All right. So why is that not working? All right. So are there any questions for that particular lecture? Are there any questions online? I have one. Is half maximal always synonymous with ED50? Yes. All right. So usually at the end of every lecture, I usually present some mock questions that are really similar to the ones I give on tests. I usually do this through turning point, but is my understanding that everybody has turning points, or just kind of, I didn't activate turning, but we'll just walk through them.
Professor, I have a quick question.
Yes, go ahead, sir.
For slide 23 and 24, the glycoproteins and the pre-pro hormones, that's the form of the hormones as it's when it's in the Golgi complex, right before it's like secreted out of the Golgi.
Hang on, let me get. Oh, sheesh. Hang on. You guys know the numbers much better than I do. Where are we at here?
23, 24.
This what you're talking about?
Yeah. So the pre-pro hormones and the glycoproteins that are shown after, those are is that the form of the hormones when they're in the Golgi complex right before they're.
So in the Golgi complex is where the glycosylation occurs. So most of the processing happens either in the Golgi process or when it gets to the secretory vesicle. That's not 100% known because it's kind of hard to isolate these. So most of the post-translational modification in the hormones is happening as it transports or transits through the Golgi apparatus. So most of the proteolytic cleavage are going to happen in there. Glycosylation, I think, mostly happens in the Golgi apparatus, but I can't completely rule out some of it happening in the secretory vesicle.
Okay. So I guess you can say the cutting of the hormones is happening anywhere from beginning of the Golgi complex to the end of the secretory vesicle when they're in there.
I say it again, I can't hear you. I did not hear you.
So my main question is the proprio hormones. Is it cut throughout the Golgi complex into the vesicle?
It can be cut in multiple ways. There's sometimes, depending on the hormone, sometimes they cut in the Golgi apparatus, sometimes it can be still cleaved in the secretory vesicle. And sometimes you can have problems where you have, if it's really secreting fast, you can secrete hormones which are not proteolytically cleaved, right? So you can secrete the pro hormone into the extracellular fluid because it's just not processed correctly. So that can happen also. It depends. So sometimes some of those proteases are in the secretory vesicle. I mean, those proteases are going to be transported along with that secretory vesicle. So it kind of depends on the hormone. Okay, I'm not going to ask you exactly any questions that'd be that specific on where it's going to be cleaved. Right?
Got it.
All right.
Okay. Thank you.
You're welcome. Any other questions online? I guess not. All right. So these are some typical types of questions that we might have on the quiz. All right. So here I'm showing you whole body glucose disposal, 0 to 100%, versus the plasma insulin concentration. Right. And I'm going to tell you that curve A is a normal human subject. Right. And I'm going to ask you which one of these other curves, B, C, D, or E, would you expect would respond to a diabetic that has both decreased sensitivity and capacity to respond to insulin. So which one of those curves would you assume that it would be? So think about it for a couple of seconds, see what your answer was, and then just, I guess anybody online can, I guess you can put it in the chat or you can just tell me what you think. All right. Anybody have an idea? Excellent. Well done. Pretty simple, huh? All right. See me? Okay, good. Dr. Berlin then. All right, so which of the following hormones?
Sorry, Professor, I have a question. So the previous question, so was it because it has lower EC50?
So decreased sensitivity and decreased capacity. The decreased capacity is dropping the maximum response down. Decreased sensitivity is shifting it to the right. So the EC50 or ED50 is shifting this way. So here's a normal hormone. It's got a, I don't know, somewhere around 1 nanomole perhaps. This one's got a EC50 somewhere around 20 or 30. So we have both a shift in the sensitivity to the right, which means it's less sensitive, decreased sensitivity, and decreased capacity because the maximum response is going down. B
And it
here,
has the
so-
lowest EC50, correct?
yes.
Okay, thank you.
So B only shows a shift in the sensitivity so it's decreased sensitivity. Curve C has an increased sensitivity, so it's completely opposite of what we'd expect. Curve D is an increase in the maximum capacity to respond, but no shift in the EC50. So E is the only one that can fit both of those parameters. Okay, I got some more things in the chat. Oh, okay, very good. All right. So which of the following hormones initiates its reactions at target cells by binding to nuclear receptors? So we went over these, so your body figures it out. Anybody have an idea? Is there more than one correct? Not that I know of. I can't hear you. All right, so it said E. And I have anybody on? Oops, sorry. That's true. So thyroid hormones, like steroid hormones, are going to bind to nuclear receptors that elicit a biological response. I can't hear you. This one, adrenocorticotropin hormone? This hormone is, yes, but it's water soluble. So it's going to bind to a extracellular cell membrane receptor and elicit a biological response. Sorry. Okay, yes. All right, so I don't know if you guys heard that. The question is the hormones from the hypothalamus bind to receptors just like hormones from endocrine glands do, right? All right, so that depends on the hormone, right? So if they're water soluble, if they're peptides, or they're made from a single amino acid like a catecholamines, they're all going to bind to receptors. They can't cross the plasma membrane. They're going to bind to receptors and elicit a biological response through that receptor signaling pathway, activating like either increases in calcium or cyclic AMP levels. All right. So anyone else? All right. So that's all right. So final question. So snare proteins are the proteins which are involved in the vesicles fusing to the plasma membrane, right? So in this example, so genetic mutations in the snare proteins inhibit calcium and triggered exocytosis. That's what we've talked about, that regulated cytosis. So of these hormones, the secretion of which of these hormones is not expected to be affected by a mutation in the snare protein? So which one of these do you believe? So which one of these would not be in a secretory vesicle?
Cortisol.
Say again?
Cortisol, C.
Yeah, true. Excellent. Well, this thing works about really poorly. All right. That's all I have for right now. Is there any other questions? There's one thing more in the chat. Okay, that's C. All right, so that's good. That down.